In the following section, a response is given to Brenden Kyer's treatise on the question, "Can genetic code come from chemicals through material processes?" A chapter of his presentation, Genetic Code, Origins of Life and Mutations. A response to Hugh Owen. Some of Brenden Kyer's key answers are the following ones. First, he answers, yes, at the very beginning, genetic code can come from material processes, but does not attempt to justify this conclusion by presenting a scientific argument on the subsequent slides. Second, although the genetic code is playing a fundamental role in all biological life, and although it represents a material structure of unimaginable complexity, having a data storage density 1 billion times larger than that of our state-of-the-art computer hard disk drives, he admits that this is not a subject I actually care all that much about. Third, instead of using the opportunity to make the apparent evidence for his yes known to the public, he advises those interested in getting to know the arguments to the literature by saying, "What I can do is I can do the research. I can point to multiple lines of scholarship. You should go and read." The only support for Kyer's yes is the presentation of summarizing quotations from the evolutionary literature, for example from Crick. The general features of the genetic code are described. It is considered that originally only a few amino acids were coded, but that most of the possible codons were fairly soon brought into use. In subsequent steps, additional amino acids were substituted when they were able to confer a selective advantage, until eventually the code became frozen in its present form. However, this statement only speculates how an already existing genetic code could have become more advanced by the principle of natural selection. The key question, how the first primitive code system came to be, is not addressed by this quotation. Another example by Barbieri. The first genetic code, in other words, was necessarily ambiguous, and its evolution took place with a mechanism that systematically reduced its ambiguity and eventually removed it. The concept of ambiguity reduction has been repeatedly mentioned in the scientific literature. Again, this is already assuming the existence of a genetic code. The quotation is not touching the topic of this chapter, how the first genetic code emerged. The closest approach given to explain where the first code came from is the reference to the so-called RNA world hypothesis. He quotes Thomas, "It seems therefore probable that the genetic code originated in an evolutionary replacement of a part of an amino acid adsorption site by the linked codon sequence of mRNA. Since it is impossible for the viewer to assess this model when no explanation of its basic contents is given, the following slides shall provide the necessary information. As DNA molecules do not have any biochemical functionality, and since proteins, which do have functionality, do not contain genetic code, another sort of highly complex macromolecule is suggested to bridge the gap. The mRNA molecule, the messenger RNA, is a copy of the DNA, which is transferred to the ribosome, the even more complex reading and construction machine in the cells, which assembles amino acids to form long polymers called proteins. RNA molecules, being working copies of the DNA strands, therefore contain genetic code. And in addition to that, they are, like proteins, able to perform some biochemical functions in the cells. For that reasons, the RNA world hypothesis assumes that in the beginning there were RNA molecules, which later evolved to form separate DNA strands. It is assumed that a primitive kind of RNA molecule contained, among others, the nucleotide bases found in DNA, and which could chemically bind the amino acids for which these bases code in the DNA. Such molecules with binding sites for just one specific amino acid are called aptamers, in the diagram illustrated in A. These aptamers are arranged in the RNA macromolecule in a well-defined order, see part B in the diagram, so that their respective amino acids are also organized in a specific sequence. Eventually, by an assumed condensation process, the amino acids will be joined to each other, forming a functional protein, part C. However, this model does not answer the decisive question how genetic code originated. It only transfers the problem from the nucleotide sequences found in the DNA to the aptamer sequences found in the RNA. How did these sequences, which represent the code for enormously complex amino acid chains, come into existence? All the arguments given in the main presentation on the origin of the genetic code are therefore also valid for the RNA code. For example, the question, where are these assumed highly evolved RNA worlds today? And the conclusion is the same as for the DNA. Natural processes are incapable of producing it. This is a fundamental problem until today, and has been summarized by Gerald Joyce, one of the leading researchers into RNA world models, in the journal Nature already in 1989. The most reasonable assumption is that life did not start with RNA. The transition to an RNA world, like the origins of life in general, is fraught with uncertainty and is plagued by a lack of experimental data.
Hello, this is Rob Stadler, and I'm responding to Brendan's statements, especially the focus on the big question of do we know how life began? Brenden, thank you very much for your response. I get it where you say that you're not so interested in the topic and you don't really want to debate or talk about these questions at all. I appreciate that. But I do recognize that this issue of do we know how life began is so foundational, because even though there are atheists with many different kinds of beliefs, as you point out, that I believe that any rational atheist, anyone who wants to claim that they're a rational atheist, would be most comfortable then if they have a naturalistic explanation for how life began. If there is no naturalistic explanation, if some supernatural agent had to be involved in getting life started, of course you could continue to be an atheist, but I wouldn't say you're a rational atheist. And that's why this is very foundational. So back in your presentation, around 35 minutes, where you hit this question, do we know how life began? You then give the correct answer of no. But I'm concerned that your explanation that follows, I believe, is very different from what science is teaching us. Your explanation that follows of why we don't know is because there are so many options, each with a plethora of evidence to describe how life got started. And so we don't know why, because there are just so many options. It's so easy to get life started that we can't know how it actually happened. And that kind of thinking, I think, is called out at 36 minutes and 30 seconds in your presentation, where you say, quote, What we do know is that there are very clear lines of scientific evidence. There have been very clear experiments that have demonstrated that actually life can begin from non-life, unquote. And then at 39 minutes, 29 seconds, you say also, quote, we 100% have been able to see both amino acids, the building blocks of life, be able to assemble on their own. And by the way, I have no problem with that part of the statement, but then it goes on, quote, and have been able to recreate situations in which we can easily see how amino acids could then form into simple single cellular organisms. That is quite concerning to me because you're espousing a belief that is completely unsupported by science. No one has ever come close to creating life in a laboratory, not even nearly close. To me, it's very analogous to saying that we have transported humans to another galaxy on a spaceship, which of course is light years away from being true. But you made those statements. And I have some sympathy for you and for what you're espousing as your beliefs, because part of the reason I think is, as you mentioned in your own talk, that science is based upon methodological naturalism, which means that science is only allowed to seek the best natural explanation. So when you ask a question about how life got started, science is then constrained to not actually answer the question of how life got started, but more constrained in saying, what is the best natural, purely natural explanation for life? That's all science is allowed to think about when it's constrained by methodological naturalism. And so therefore, science with that constraint doesn't actually seek truth. It only seeks the best natural explanation. And I can tell you from 30 years of publishing in scientific manuscripts that there is inherently bias in all of publications for positive results. And so if you're only allowed to speak about natural explanations and you're very predominantly publishing only positive results or allowed to get into journals, I can see where there's a strong bias there that you have to publish positive, natural explanations for things. And of course, the media has a way of hyping that up and amplifying it. And as a result, many, many people are misled by media, by what they hear, into thinking that, in fact, life has actually been created in labs, that scientists have made this great progress. And the papers that you cite largely are a few science journals, but a lot of them are just media science summaries in popular media. And I find them to be very misleading. If you actually dig into the science, and especially if you review the presentation that I started with, that 20-minute presentation, you'll find that there are so many gaps. And the scientific papers you cite also tend to refuse to list the limitations of what they come up with, and they only show the positive stuff, and it misleads a lot of people. And another big part of why I think people believe as you do is a very strong bias. In your own presentation at 12 minutes, 30 seconds, I think you very correctly stress the importance of avoiding bias when you say that avoiding preconceived ideas is the right way to find the truth. And then at 15 and a half minutes, I know you were at the time speaking about religious people, but you said, quote, if you begin from the point where I already know that my faith tradition is the correct one, and then go and look for evidence, what you're really doing is looking for evidence to confirm your pre-existing bias, and you disprove others' assertions. That is not good science. I completely agree with you there. I just also think that it can be, your own words should be applied to your own beliefs in that case. And I fear that your own bias is making it difficult for you to see the truth in this situation. In fact, I've worked with a lot of people in situations like this, and I find that they often subordinate actual science, actual evidence, to their own beliefs. And in those cases, I find that discussing science, discussing the evidence is actually a complete waste of time. It's irrelevant in the sense because they've already made up their minds and there's no point to discuss the evidence. So if I'm being unfair and you're thinking otherwise, I would certainly invite you to contact me at my website, which is scientificevolution.com. And I'd be happy to continue this conversation. Again, thank you for putting up with this discussion and for keeping with it. And I wish you the best. Thank you.
Again, and I am here to answer some of the points that were raised by Mr. Kyer in his portion of the recent debate. So in the first place, I'd like to point out that I find it rather ironic that Mr. Kyer puts such tremendous weight on peer-reviewed papers and how important the peer review process is, and then uses a blog from Big Think and a Reddit thread to send you, his listeners, to prove his points. So 0 points for consistency on that part of the assignment. I'm going to talk just about a couple of the points that he raised because I honestly rebutted a lot of what he said in my previous presentation, but we'll deal with a little bit with mutations. So he's talking about mutations, quote unquote, adding new information to the genome, but ignoring, again, the problem that we have to go from God is good to question 6 of the Summa, or actually 3 or 4 times that. So we're talking about mutations that build information in the genome, and most of the ones that he mentions, like eye color and tetrachromacy and lactase persistence, don't build information and don't actually positively impact fitness either. And a number of the ones, especially from the Big Think article, that are supposedly beneficial mutations are, once again, examples of mutations that are actually harmful. And evolutionists want to explain this away by saying, well, fitness is totally relative and you creationists just don't get that. Fitness isn't totally relative. Remember, it's not completely fungible. So if I've lost, function in a very important enzyme that I need in order to survive in areas where there are oxygen, then I can't survive in areas where there are oxygen, which is everywhere. Okay, so not all changes to fitness are fungible. Blindness in cavefish is an example evolutionists like to use a lot. And they say, well, now the fish doesn't have eyes, so it's in less danger of dying from an infection by damaging a very sensitive organism. But it also has a problem. If its environment changes and its little cave pond dries up, it can't just swim out into the river and exist very well. It's going to get eaten by a big predator pretty quickly, and that's going to be game over. End of that mutation. So you can't just claim that all fitness is totally relative. That fits with the worldview of moral relativism, but it doesn't fit with reality. Reality is harsher than that. You do actually need certain things in order to survive in the world. If those things get damaged, it's game over. Okay. So in these examples from Big Think, there's 3 mutations. One is supposed to reduce bad cholesterol. It actually just reduces a very specific enzyme that's involved in very important regulation of the homeostasis of cholesterol, which you need for your cell membranes and also for hormone production. It reduces the function of that enzyme to the same function as vitamin C, which is not a benefit. There's dozens, hundreds of simple chemicals that could perform that function for you. The PCSK9 mutation that's supposed to reduce heart disease increases non-alcoholic fatty liver disease, and the LRP5, which increases bone density, is supposed to protect against osteoporosis, can actually increase bone density to the point that it causes hearing loss and decreases circulation to your brain. So relative fitness, barely. Okay. It's just be very, very, very careful when you see people claiming that supposed beneficial mutations, and especially when you see them pulling them off the blog. Go and look at the actual mutations in the actual papers in the actual journals. Okay. So unfortunately, it's common for evolutionists to just assume that there are beneficial mutations and then do a very, very, very basic and not well-researched search to find a few and throw them up and say, look, beneficial mutations. But they're operating under this assumption that, well, since they know evolution is true, then they know there must have been beneficial mutations that accumulate. But this is actually just circular reasoning. And it's a huge bias that's inherent in the supposedly unbiased methods they're using, right? So Mr. Kyer wanted to make a big point about methodological naturalism being not biased, but that statement is a profound misunderstanding of both bias and philosophy. So for a really great explanation on this, much better than I could give, I recommend going to Michael Knowles' channel on YouTube and looking up his talk, I'm Fine with Being Called Anti-Science. He does a great job of explaining the bias inherent in methodological naturalism, because you can't approach anything without bias. If you have a worldview, you have bias, period. The atheist worldview is not unbiased, and the naturalist worldview is not unbiased either. So one of the other points that I have issue with Mr. Kyer's debate portion is that he gave a complete misrepresentation of junk DNA. I don't know what he's reading that made him think that we've done experiments where we've knocked out massive portions of genomes and organisms that survived just fine. But I'd love to see it, because that is absolutely not my experience. My experience has been that when we have knocked out, I've worked with knockout organisms in the laboratory. When we knock out stuff, even in supposedly junk DNA regions, you have problems with functionality. Just like I talked about that example with the worm that couldn't defecate and couldn't lay its eggs because of a change in an intron, which is supposed to be junk DNA. So for an actual explanation of why junk DNA isn't a real concept, I again refer you to Jonathan Wells' book, The Myth of Junk DNA. And because the experiments that are being done, especially within code, as I referenced before, are showing again and again and again, this DNA is being transcribed, it's being translated, it's performing regulatory functions. There's no such thing as junk DNA. The science shows us there's no such thing as junk DNA. All the stuff is published in peer-reviewed papers too. And then the last thing that I have issue with Mr. Kyer's comments is the way that he treated Dr. Sanford, which was both uncharitable and just flat wrong. So there are examples of both error catastrophe and genetic entropy in real life. Dr. Sanford and Robert Carter, who I personally know, have published, and he's a population geneticist. This man is no dunce at this stuff. He's also a man of tremendous integrity. They published a paper in a peer-reviewed journal looking at influenza genomes and showing that genetic entropy occurred in a population. And there have been some questions raised about that research, and Sanford and Carter have done a great job of answering those questions on creation.com. You can look it up. And again, that's peer-reviewed research. Then you can look at natural populations like cheetahs and snow leopards, things that are on the verge of extinction. They've gone through population bottlenecks. Their populations are imploding because of genetic entropy and genetic decay. They've accumulated so many negative mutations that they can't back cross to a mate that has a sufficient amount of correct alleles for the genes that they have really negative mutations in. And you're getting a problem where kittens are dying in utero, or they're dying, shortly after being born, because they simply can't survive because the load of detrimental mutations. So you have a mother that's wasting all this time and energy on gestating something that can't survive, and she's missed another breeding season, and your population just plummets. And there's kind of no way back after a certain point. You just hit extinction. Again, we see this in real life. We see this in the real world. So it's super spurious to say there's no examples of this. The T7 paper that Kyer uses, where he claims represents, look, science has shown that genetic entropy isn't true, and that as mutations accumulate, you get increases in fitness to the population. He obviously didn't read that paper, because when you read the paper and not just the abstract, you see that the scientists themselves have to wrestle with this problem, that while they assigned a number to fitness that they claim has gone up over the 200 generations of the experiment, the burst size of the virus has gone down by 80%. That means that every time the virus replicates, there's 1/5 the number of virus particles being produced at each round of replication as there were before. So you can't tell me that you've increased fitness, because fitness is being measured by reproductive success, in a population that's only producing 1/5 of what it used to produce. That's inherently contradictory. And the guys who authored the paper wanted to wave it away mathematically somehow, but you can't wave things away mathematically. And Kyer has a good point about that. He brings up Mendel's accountant, which he doesn't reference by name, which is a program that Sanford and Carter put together, and they use it to model genetic entropy. Well, evolutionists have programs they use to model, their ideas of the way that things go. And all of those programs are going to give you the results concomitant with what you expect, because the parameters that you put in are going to determine the outcomes that you get. So if you think that the original parameters were this way, you're going to put them in and you're going to get a certain outcome. If you think they were the other way, you're going to put them in, you're going to get a different outcome. So let's just throw mathematical models out on both sides, please, because I think they're very easy to manipulate, and a single real world experiment that shows that they're defunct. So we have real world experiments that show genetic entropy is happening. We don't need Mendel's account, right? And then, the last issue that I have is, Kyer gives a sentence explanation of genetic entropy and then says this is totally not nuanced and all of these things. And of course it's not nuanced, because Kyer himself has oversimplified Sanford's very nuanced argument. So you need to read Sanford's book, Genetic Entropy, to actually understand the argument that he's making and all of the incredible nuances. I couldn't go through it in 2 hours if you gave them to me, and I only have 10 minutes. So read that book, read his articles on creation.com where he rebuts them. And keep in mind that this Reddit thread that Brenden is sending you to is not peer reviewed. So nobody's checking the work of these PhD scientists. And these PhD scientists are primarily not population geneticists. So they don't necessarily know what they're talking about. And they don't belong in a field to be able to actually critically evaluate Sanford's claims. So I do strongly encourage you, after you've watched this debate, to do your own research, to read the relevant stuff, and keep looking for the answers to the questions that you have, because there's so much out there to learn. It's actually amazing. And don't take my word for it. Do your own homework, and I know that you will come to an excellent conclusion. May God bless you.
In the name of the Father, and of the Son, and of the Holy Ghost. Amen. I'm going to conclude our response to Brenden Kyer on the origin of coded information, life, and the possibility of new functional information arising through material processes in this 5-minute conclusion. We've seen that all of our experience in nature and in the laboratory tells us that functional information can only come from an intelligence and a free will. Take the example of a group of child cousins on holiday who invent a code to announce the coming of grown-ups. 1 clap equals Aunt Judy, 2 claps equals Uncle Don. These children must have a mind to say X equals Y, and a free will to say that X equals Y and not Z. Moreover, the density and complexity of the functional information in the cells of all living things far surpasses the capabilities of the most brilliant human intellects. We now know that DNA sequences contain sentences that can be read in one direction to give one set of instructions, in the opposite direction to give another set of instructions. Then every third letter can be read to produce a third meaningful set of instructions, and a fourth set of instructions can be derived from the same DNA sequence by translating it into another language. This kind of coordinated complexity always originates with a mind and a free will. There are no exceptions. The chemicals that make up nucleotides do not spontaneously organize themselves into meaningful letters, just as amino acids do not spontaneously organize themselves into the building blocks of living things. Moreover, while reason tells us that a tremendous mind and will must be responsible for the designed complexity of living things, in the Catholic Church we have overwhelming evidence that the Creator brings life from non-life every day in the Holy Eucharist. At the Last Supper, before he died for us, our Lord Jesus Christ took bread and wine in his sacred hands and pronounced the words, "This is my body and this is my blood," over them, thus changing ordinary bread and wine into his living body, blood, soul, and divinity under the appearance of bread and wine. 153 church-approved Eucharistic miracles prove that our Lord continues to bring life from non-life in every Holy Eucharist. For example, on August 15, 1996, in a parish in Buenos Aires, a parishioner received a consecrated host in his hands and dropped it on the floor. The parish priest, following the rules of the church, put the host in a container filled with water and left it in the tabernacle so that the host would dissolve. On August 26th, the tabernacle was reopened to remove the fallen host, and it was found that the host had not dissolved and had some reddish stains. The famous histopathologist Dr. Robert Lawrence examined the host. He concluded that the portion of the host that he examined was the tissue of an inflamed heart, which meant that the person to whom it belonged must have been in great pain. Later, to remove any doubt, the sample was given to a leading expert in cardiac pathology, Professor Frederick Zugibe. The professor didn't know that the sample was from a consecrated host. After studying it, he said, "The sample you brought me is a heart muscle, more precisely the left ventricle." Dr. Zugibe then asked Dr. Lawrence whose sample it was. And when Dr. Lawrence told him that it was from a consecrated host, Dr. Zugibe said, "Doctor, when you brought me that sample, that heart was alive." To deny that this Eucharistic miracle demonstrates God's supernatural power to bring forth divine human life from non-living bread we would have to assert that Dr. Lawrence replaced the bleeding host of Buenos Aires with a sliver of a heart from someone he murdered just as he entered Dr. Zagive's presence. One more striking example of God's power to bring life from non-life can be found in the many documented resurrection miracles recorded in the canonization processes of the saints. In the streets of Bhutan, India, St. Francis Xavier met a funeral procession bearing the body of a youth who had just died. The body had been kept for 24 hours wrapped in a shroud. The saint knelt down, raised his eyes to heaven, and prayed to God for the lad's life. The youth rose up alive, and Francis gave him to his parents in good health. At another time, St. Francis was preaching in Travancore in India. Now it happened that a man had been buried in the church the day before. St. Francis stopped preaching and prayed to God to honor the blood in the name of his son and to soften the hearts of the congregation. Then he directed a few men to open the nearby grave of the man who had been buried the day before. When they opened the tomb and brought out the body, it was already giving off a stench. It was nothing but a disorganized mess of chemicals. On Francis' orders, they tore apart the shroud to find the body already beginning to putrefy. Francis fell on his knees, made a short prayer, and commanded the dead man in the name of the living God to arise. And the man arose alive and in perfect health. These two miracles were accepted by the auditors of the Roman Rota as resting on incontrovertible evidence from two witnesses, as well as from one of the dead men himself. These great miracles led almost the entire kingdom to become Christians within a few months. I will leave it to our friends in the audience to decide whether it is more reasonable to deny all of these facts from a position of dogmatic materialism, or to acknowledge that the existence of coded information and designed complexity in all living things points to their source in an exalted intellect and will, and that the Eucharistic and resurrection miracles of the Catholic Church demonstrate that the same exalted intellect and divine will bring life from non-life every day in the Holy Eucharist, and even raise the dead at the prayers of God's saints when they ask according to his most holy will.